Hello, learners. Welcome to our lecture on Hidden Facts series in biology. We'll be looking at cell structure, but before we dive into cell structure properly, I want to take us briefly on cell theory. What does the cell theory state? The cell theory states as follows: 1. All living things are made up of a cell or cells. 2. The cell is the structural and functional unit of all living things. 3. New cells arise from pre-existing cells by cell division. Let us look at the scientists that contributed to the cell theory. First one we will look at is Robert Hooke. Robert Hooke, a British scientist, in 1665 discovered the honeycomb structure of the cell. He is seen as the father of cells. This is because he is the first person that discovered the honeycomb structure of cells. There was, however, a limitation to Robert Hooke's work. He did not realize that a cell has content, and that was because he did not examine a thin slice of living plant or animal. Now we'll be looking at another contributor to the cell theory, Félix Dujardin. In 1835, Dujardin, a French biologist, examined thin slice of living plant under much improved microscope and discovered that the cells have content. He named the content sarcode, which was later named protoplasm by Purkinje in 1839. We see that Félix Dujardin's work was an improvement of Robert Hooke's work because he examined the thin slice of living plant, unlike Hooke that did not examine the thin slice of living plant or animal. The third person we'll be looking at is Purkinje. And we have already seen that Purkinje was the first scientist to name the cell content as protoplasm, and he did that in 1839. Now, the next person we'll be looking at is Matthias Schleiden. In 1838, Matthias Schleiden, a German botanist, he carried out an extensive study on the structure of plants, and he concluded that plants were composed of cells. Another contributor to the cell theory is Theodor Schwann. In 1839, Theodor Schwann, who is a German zoologist, examined bits of animals under a microscope and observed that animals were composed of cells. The next person we'll be looking at is Rudolf Virchow. In 1855, Rudolf Virchow, a German biologist, stated that all cells come from cells. That is, cell arises from pre-existing cells by cell division. Now we'll be looking at cell structures and functions. First of all, I will be showing you the diagram of a plant cell and an animal cell side by side. So we can see the plant cell over there, and we can also see the typical animal cell over there. So to understand the structures that are composed in the plant and animal cell, we need a microscope to do that. But what I want you to know is that every cell is composed of protoplasm, and protoplasm can be divided into two main parts: the cytoplasm and the nucleus. Each cell, either plant or animal cell, is bounded by a thin membrane, while the cytoplasm that is contained in that cell is fluid-like material, and this fluid-like material consists of many organelles, and these organelles are known as cytoplasmic organelles. They include the lysosome, which you see in the diagram there labeled. Also present is the endoplasmic reticulum. Endoplasmic reticulum is another word cytoplasmic organelle. You will still see it labeled there. You can also see the mitochondria and the vacuoles with many other organelles. The nucleus itself is bounded by a nuclear membrane, and this nucleus consists of chromosomes and nucleolus. Another thing I want you to take note is that the animal cell, in addition to the organelles I've mentioned, contains centrosome, while particular to the plant cells are the cellulose cell wall, which you will see is absent in the animal cell. Also, starch granules are present in the plant cells, and plastids such as chloroplasts can only be seen in plant cell and not in animal cell. At the end of this lecture, we'll be looking at the difference of plant and animal cell specifically. But right about now, we'll be describing the components of the cell organelles in detail. The first component of the cell we'll be looking at is the cell wall. The cell wall is the non-living outer boundary of the cell. It is made up of cellulose. It has tiny pores, or what we can call openings, through which nutrients pass from one cell to another. The cell wall is absent in animal cells. What is the function of the cell wall? The function include: number one, it gives rigidity to cell and the plant as a whole. Two, it allows free passage of materials. Now, let's look at the cell membrane, which is also known as the plasma membrane. The cell membrane is a thin, flexible layer that surrounds the entire cytoplasm. It prevents the cell content from escaping, and it also controls the material which are allowed to enter or leave the cell. The cell membrane is selectively permeable. One thing I want you to know is that the cell membrane is present in both plant and animal cells, and this is unlike the cell wall, which is present in only the plant cell. The next structure or organelle we'll be looking at is the endoplasmic reticulum. The endoplasmic reticulum is a system of canal that is found abundantly in the cytoplasm of cell. There are two types of endoplasmic reticulum. We have the rough and the smooth. Take note, ribosomes are attached to the surface of the rough endoplasmic reticulum. Due to the parent of the ribosomes, that is why the rough endoplasmic reticulum is known as rough endoplasmic reticulum. It is the ribosomes that makes it rough. Now we'll be looking at the functions of the endoplasmic reticulum. First, we'll look at the function of the rough endoplasmic reticulum. One, they help in formation of enzymes and protein. Two, they transport metabolic products within cytoplasm and nucleus. Three, they assist in the formation of nuclear membrane during cell division and during nuclear division. Four, they interconnect the organelles of the cell. And five, they provide surface for the attachment of ribosomes. Now, let's take a look at the functions of the smooth endoplasmic reticulum. The first one is that the smooth endoplasmic reticulum is responsible for making lipids, that is, fat molecules. Two, the enzymes of the smooth endoplasmic reticulum, that is, the enzymes that are present in the smooth endoplasmic reticulum in the liver, detoxificate drugs such as morphine and codeine. By detoxification, we mean that the enzymes of the smooth endoplasmic reticulum present in the liver help to render the toxic nature or the toxic effects of drugs such as morphine and codeine less toxic. That is, these drugs will become less harmful from their side effects. The next component we will be looking at is ribosomes. Ribosomes are round bodies that are found free in the cytoplasm or attached to endoplasmic reticulum. In this case, it is the rough endoplasmic reticulum, like what we discussed earlier. Now, what is the function of ribosomes? Ribosomes are the sites of protein synthesis. That is, protein are synthesized in the ribosomes. 2. They also contain large amounts of ribonucleic acid. Ribonucleic acid is known as RNA. Now we'll be looking at another cell organelle. This is the mitochondria. These are tiny rod-shaped bodies. They can be described as sausage-shaped. They are surrounded by double membrane layer. The mitochondria are more concentrated in very active cells such as the liver cells. What is the function of the mitochondria? The function of the mitochondria includes 1. They are the center for cellular respiration, in which food substances are oxidized to release energy for the activities of the cells. That is, cellular respiration takes place in the mitochondria, and this cellular respiration is needed for the generation of energy. Hence, they are referred to as the powerhouse of the cell. The mitochondria is the powerhouse of the cell. 2. The mitochondria contains enzymes and the oxyribonucleic acid DNA. These enzymes carry out oxidative phosphorylation. By oxidative phosphorylation, we mean addition of phosphate. They carry out oxidative phosphorylation of adenosine diphosphate, and when you add a phosphate group to adenosine diphosphate, you get adenosine triphosphate, also known as what? ATP. The enzymes present in the mitochondria carry out oxidative phosphorylation of adenosine diphosphate to adenosine triphosphate, which is known as ATP. The DNA helps to code the synthesis of protein in mitochondrial membranes. Now we'll be looking at another cell structure known as the lysosomes. Lysosomes are small round sacs that contain digestive enzymes which break down structure and substance. It's sometimes referred to as suicide bags. What are the major functions of the lysosome? They include: number 1, they destroy one part of the cells by discharging enzymes into them and clearing the area for a new healthy cell to grow. So they clear the area and a new healthy cell will grow. And how do they do that? By discharging enzymes into those one parts. 2, they discharge enzymes to degenerated tissues, causing the breakdown of the cells. 3, enzyme released by lysosome destroy bacterial cells. Now we'll be looking at the Golgi apparatus or Golgi bodies. The Golgi bodies are found in group as flat tubes or threads. They are absent in mature sperms and red blood cells. What are the functions of the Golgi bodies? 1, they help in distributing proteins made by the cells. 2, they help in the manufacturing of lysosomes. 3. They help in the formation of membrane of endoplasmic reticulum and production of cellulose cell wall. To summarize, the function of the Golgi bodies is in the synthesis, packaging, and the distribution of materials. We'll be looking at the central now. The central are found mostly in animals. They are usually absent in plant cells except for some few algae and fungi. What is the function of the central? The function include 1. They provide spindle fibers to which chromosomes are attached during cell division. 2. They help in the formation of cilia and flagella. Cilia and flagella are organs of locomotion. Next, we'll be looking at plastids. And plastids are usually found in plant cells. Plastids in plant cells. These plastids are of different type. The first one is the chloroplast. What is the chloroplast? The chloroplast occurs only in green plant. That is, we see them only in green plant. It contains a pigment called chlorophyll. The chloroplast is the site of photosynthesis. The next plastids we can see in plant cells is the leucoplast. Leuco means white or colorless. Hence, these are colorless plastids, mainly for storage of starch and liquids. The next plastid is the chromoplast. The chromoplast are plastids that contain different color. For example, when we look at the petals of flower, we see different colors. The colors of petals of flowers are due to these plastids. The next cell organelle we will be looking at is the vacuoles. Vacuoles are mainly found in plant cells, but they might still be seen in animal cells. But the difference is that when they are found in plant cells, they are large. That is, they are very large in plant cells. But when we see them in animal cells, they are usually small, but most times they are absent. That is, vacuoles present in animal cells are small, but most times they are absent. What is a vacuole? A vacuole is usually surrounded by a membrane, and this membrane is called a tonoplast. Vacuole is usually filled with water, minerals, salt, sugar, and pigments. These pigments, they are called the cell sap. Now we'll be looking at the function of the vacuole. The first one is that the cell sap is osmotic in function. When we say osmotic in function, what do we mean? We mean that the cell sap helps to maintain the balance between water and intracellular fluids. The next function of the vacuole is that it serves as a storage for nutrients and waste products. The next function is that the vacuole can contain food reserve such as sucrose and minerals, and these food reserves will be available when the organism needs it or when the plant needs it. The next function is that the vacuole can contain tannins and alkaloids. These tannins and alkaloids have function. The tannin and alkaloids, they prevent herbivores from eating plants. That is to say, those plants that contain these chemicals, tannin and alkaloids, they won't be able to be consumed by the herbivores. That is, tannin and alkaloid prevent herbivorous animals from eating plants and animals that have these chemicals. The next function is that the vacuoles sometimes contain color pigments. This color pigment is known as the anthocyanin, and this anthocyanin gives coloration to flowers, fruits, leaves, and buds. Now, we'll be talking about the nucleus. Before we talk about the nucleus, I want to make a little recap. Remember, I said that the cell is composed of protoplasm, and the protoplasm consists of the cytoplasm and the nucleus. And this protoplasm is surrounded by what? A membrane. The organelles we have looked at thus far are the organelles contained in the cytoplasm. Now, we will be looking at the nucleus. Let's talk about its structure. The nucleus is bounded by a thin nuclear membrane which separates it from the cytoplasm. So the nuclear membrane separates the nucleus from the other content of the protoplasm, which is the cytoplasm. Now, this nuclear membrane has many tiny pores, which can also be called pits. These pores or pits allow substances to go in and out of the nucleus. The nucleus contains a small spherical body, and this small spherical body is known as the nucleolus. It also contains chromatin materials which condense during cell division, and when they condense, they form the chromosomes. These chromosomes are hereditary units. The nucleus also contains a fluid, and the fluid inside the nucleus is known as the nucleoplasm. The chromosomes, which we said earlier, are made up of DNA, and that is deoxyribonucleic acid. It is important for you to know that DNA does not pass out of the nucleus, and also important for you to know that the red blood cell in man lacks nucleus. Take note of that fact. The red blood cell in man lacks nucleus. Now, what is the function of the nucleus? It includes, number one, it controls directly or indirectly most of the activities of a living cell. 2. The nucleus carries chromosomes on which hereditary materials, that is genes, are coded. 3. The DNA in chromosomes gives information for the manufacture of the proteins in the cell. 4. The nucleus produces several kinds of RNA. RNA means what? Ribonucleic acid, which are passed out of the nucleus to the cytoplasm to manufacture protein. Now we see that it is the RNA that actually leaves the nucleus, not the DNA, like I have mentioned earlier. So this brings us to the end of the cell structures and functions. Now, having looked at the different cell structures and their functions, if you remember earlier, I showed you a diagram depicting the plant cell and the animal cell. Now, I will be showing you the differences between the plant and animal cell. I will kindly refer you back to the diagram I showed you earlier. You can check it out and use it to consolidate your understanding about this concept. So we'll be drawing a table. On the left hand side, we'll have plant cell, and on the right hand side, we'll have animal cell. Number one difference is that in a plant cell, cellulose cell wall is present, but for animal cell, cellulose cell wall is absent. 2. In a plant cell, chloroplast is present. In an animal cell, chloroplast is absent. 3. In a plant cell, a large central vacuole is present, while in an animal cell, small vacuole may be present or vacuole may be absent. 4. In a plant cell, centrioles are absent, but in an animal cell, there is the presence of centriole. 5. In a plant cell, the cytoplasm is pushed towards the edge of the cell wall. Cytoplasm is pushed towards the edge of the wall, but in animal cell, cytoplasm spread throughout the cell. 6. In a plant cell, starch granules are present, but in an animal cell, glycogen granules are present. 7. The shape of a plant cell is usually definite and rectangular, while for that of an animal cell, the shape is spherical and not definite. That is, it has a spherical and no definite shape. Now, we will be looking at some examples. Our first example, it says the part of the cell that stores acid is A. Nucleolus, B. Nuclear membrane, C. Chromosome, D. Nuclear pore. From our teaching, we explained that the chromosomes are made up of DNA, and DNA means deoxyribonucleic acid. That means the answer to this our question is C, chromosome. For our next question, it says animal cells are different from plant cells because animal cells have A. Definite cell wall, B. Large vacuole, C. Centrioles, D. Chloroplast. From our teachings, we see that option A, the definite cell wall, option B, large vacuoles, and option D, chloroplast, are specific features of the plant cell. The odd option there is C, which is centrioles. Centrioles are particular to animal cells. It is found only in animal cells and play a role during cell division. The answer is centrioles. The next question, it says, which of the following structures controls the activities of the living cells? A. Nucleus, B. Centrosome, C. Chloroplast, D. Golgi body, E. Mitochondria. The activities of the cell is controlled by the nucleus, option A. The nucleus is regarded as more or less the brain of the cell. All other options are wrong. So our next question, most of the energy in the cell is produced in the A. Mitochondrion, B. Lysosome, C. Plastid, D. Golgi apparatus, E. Cytoplasm. Referring back to our teaching, we saw that the mitochondrion is the organelle which is known as the powerhouse of the cell. It is the organelle where cellular respiration takes place, and cellular respiration helps in the production of what? Energy. So the answer is mitochondrion. Now to our next question. The structure in the cell that controls the movement of substances in and out of the cell is the A. Cytoplasmic membrane, B. Nuclear membrane, C. The cytoplasm, D. The protoplasm, E. Endoplasm. The correct option is A. The cytoplasmic membrane. The cytoplasmic membrane is also known as the cell membrane or plasmalemma or plasma membrane. It is a thin flexible layer that surrounds the entire cytoplasm, and it controls the movement of materials in and out of the cell. The next question. The first scientist to describe the cell was A. Theodore Schwann, B. Felix Dujardin, C. Robert Hooke, D. Charles Darwin, E. Matthias Sliding. From our teachings, the answer to this question is C. Robert Hooke, who in 1665 described the cell. Robert Hooke was a British scientist. So our next question, it says the structure that produces ATP is labeled A. VII, B. V, C. VI, D. I. If you refer back to the diagram I showed you earlier during the course of our lecture, you will easily find out that The part labeled VII is the mitochondria, and the mitochondria is responsible for producing ATP. So this makes our answer to be VII. And also remember that ATP is the energy currency of the cell. The second question on this diagram says the structure labeled V is the A. Cell wall, B. Nucleus, C. Endoplasmic reticulum, D. Mitochondria. If you still refer back to the diagram, you will see that V is the cell wall. This diagram is that of a typical plant cell, and plant cell has cell wall. So this brings our answer to what? Cell wall. Now our next question says, list 3 ways in which a typical plant cell differs from a typical animal cell. Then our answer. From our lecture series, we measured several ways we can differentiate a plant cell from an animal cell, but we'll just take 3 of them. Number 1 is that the cell wall in plant cell is made of cellulose, but the animal cell, the cellulose cell wall is absent. Number 2, the plant cell possesses chloroplast, but chloroplast is absent in animal cell. The third difference is that plant cell lack centrioles, but centrioles are present in animal cells. Our next question says, one of the differences between plant and animal cells is that in plants, A. cells have less distinct outline, but animal cells have distinct outline. B. Cell wall is made of cellulose, but animal cell wall is made of chitin or chitin. C. Large vacuoles are absent but present in animals. D. Plastids are absent but present in animals. E. Carbohydrates are stored as starch but as glycogen in animals. Now to our answer. If you check our differences, you will see we mentioned that starch granules are present in plant cells, while glycogen granules are present in animal cells. That is, plant stores carbohydrates as starch and animal store carbohydrates as glycogen. This brings our answer to E. Carbohydrates are stored as starch in plants but as glycogen in animals. These are the few examples we can take. We will be taking some exercises, and these exercises were also gotten from our hidden facts in biology textbook. You see that in page 57 of the textbook. Try your hands out on them, and if you will be needing more exercises, you can get the hard copy of the book, for there are more exercises there, and the answers to these exercises are at the back of the textbook. Thank you.